Okay, I think I'm going to get started. So, second episode with me today. Before the break, I was talking about genomic sequence, and I think as I said, I agree with Greg. It's really a good starting point. But there are a lot of things that just looking at the genomic sequence really won't be able to tell you. And in fact, the genome sequence just tells you what the parts list is. Now you have to figure out how the cells use that to do what they're doing. So I'm going to spend the next hour or so showing you some technologies that we've been using for RNA-seq, and then hopefully showing you a few examples of where that's told us a few interesting things about how Leishmania works. So just to remind you that transcription and RNA processing are a little bit different in trypanosomatids than in other organisms, and in that they have polysystronic transcription. Transcription starts generally in these divergent strand switch regions, usually in runs of Gs actually in these strand switch regions, and proceeds polysystronically transcribing, sometimes hundreds of genes until it ends usually, but not always, in a convergent strand switch region, which often has a tRNA gene involved with it. The control of where transcription initiation starts and ends involves epigenetic elements, including the chromatin, modified chromatin. There's acetylated H3 and H4 that are associated with transcription initiation sites, and H3 and H4 variants at the termination sites. And then also base J is associated with termination sites as well. And I'll talk a little bit about the base J stuff. So while this transcript is being made, it is also being processed by the splicing machinery. And the splicing machinery actually associates with the polymerase. So this enormous complex comes together to both make the RNA, and as it's being made, it's being processed into individual messenger RNAs. And so what that process involves is the addition of a 39 nucleotide splice leader sequence that's transcribed in Leishmania from chromosome 2. That is spliced by trans-splicing onto the 5' end of all messenger RNAs. And that is coordinated with the polyadenylation of the upstream messenger RNA. So that at the end of the day, what you get is an RNA that has a polyA tail on one end and a splice leader site on the other end. And the splice leader site is present on all messenger RNAs, and that actually is particularly useful for molecular biology, as we'll see in a moment. What's interesting, though, is even though these are all made from the same RNA precursor, so therefore they are transcribed at exactly the same rate, and in fact there doesn't seem to be any real difference in the rate of transcription between different chromosomes in Leishmania, with the possible exception of chromosome 31 maybe. Even though they start off with exactly the same RNA levels, by the time they are present as mature messenger RNAs, the stable RNA level of each individual messenger RNAs can vary tremendously. And so that two adjacent genes can have 100 or 1,000-fold difference in their steady state levels. So there's a lot of control of RNA levels that can happen during this splicing stage, and then after the mature messenger RNA has been made, there are elements in the 3' UTR usually which regulate the stability of that RNA. That can also be regulated developmentally and in response to stimuli, and so we'll see how that works later on. Okay. So, obviously what we want to do is we want to see how these changes in messenger RNA levels or in transcription rate can be controlled and under different conditions. And there are a number of techniques that are available to analyze transcription that I've outlined up here that are used in essentially all organisms. And I'll just very, very briefly go through some of those and dismiss most of them. So those of you who have been around as long as I have will remember that when you wanted to look at RNA levels, you did northern blots, right? So you looked at a single gel and you measured the steady state level of the RNA. Northern blots were what you did before RT-PCR was invented. I actually like northern blots because not only do you get a measurement of the level, but you get the size of the RNA. So you get more information than you will get from an RT-PCR experiment. They're a lot more work though, and you can only really do them at one at a time. And they involve, at least in the old days, used to involve hybridization with radioactive probes. So not really very many people do northern blots anymore. Quantitative real-time PCR is probably what many of you have done now, and the advantage of that is that it's a little more quantitative. You can multiplex it, and you've got a variety of different probes that you can use. I'm not a particularly large fan of quantitative RT-PCR because I believe that there's too many mathematical manipulations that go into measuring the levels. But nevertheless, it's quite useful from time to time. And you can measure perhaps 10 genes at the same time. After that, there was a time in the, well, I don't know, mid-2000s or late 2000s when microarrays were all the rage because you could basically use a microarray to measure the steady state levels for all of the genes within a genome. And generally what will happen is that you will immobilize a DNA fragment, usually an oligo these days, to a platform, and often you'll have, in fact, it's preferable to have more than one probe per gene. And then you make a cDNA from the RNA, and you label that, and you hybridize it to your array. And then generally, you can use two different labels, and so you can compare the levels of RNA under two different conditions. And so you end up generating a heat map like this, where everything that's in red is upregulated relative to in that sample relative to the reference, and everything that's in green is downregulated, and then everything that's black is sort of the same. So you generate these heat maps, which really tell you quite a lot. And the big advantage is that you can essentially look at all of the genes in the genome at once. The problem is that it requires specially designed chips, and for organisms such as Leishmania, they don't have very many people working on them. You can't just go buy them off the shelf. You had to spend many tens of thousands of dollars to buy the chip. And so it was a relatively expensive undertaking. Starting 4 or 5 years ago, that was really replaced by RNA sequencing, which is what I'll be talking about most of the time today, where you make cDNAs, and then instead of hybridizing, you just sequence them using usually an Illumina platform, and you generate tens or hundreds of millions of reads. And so one of the big advantages of that is you don't actually need to know what the genes are, because you'll find out what they are by sequencing them. Now, you do need to have, to use this properly, you do need to have a reference genome. So you do need to know something about it, but you don't strictly need to know the position of each RNA. And in fact, the experiment will tell you where the RNAs are. And you can use this information to map the transcription start sites. You can map alternative splicing. You can look for small RNAs, et cetera, in addition to measuring the levels of each of them. The other advantage is you get a very quantitative and digital signal. So the dynamic range that you get from RNA sequencing is in the order of 5 orders of magnitude, whereas the dynamic range of a microarray is about 2 orders of magnitude. If you do a microarray experiment, it's very unusual that you ever see any gene that changes by more than 100-fold. In this, you can see changes of 10,000-fold. Not that in Leishmania you ever see changes of that magnitude. I mean, in Leishmania, RNA levels only change by 100-fold for just a few genes. in the TRIPs, on the other hand, you do see 1,000 or 10,000-fold changes in a few of the genes. There's another technology that's even newer called Nanostring, which has the advantage in that, which I'll come to in a moment, but essentially it's sort of like a microarray, but it's digital. And the way it works is that for each gene, you design 2 probes. And one of the probes is called the capture probe. You design so that it matches a unique sequence on your messenger RNA. And it contains, on the other end of it, it contains some repeat sequences that serve to capture it and capture it on a glass slide so that you can then have those lying down on a glass slide, and then when you put the RNA on top of it, that will be captured by this region here. There's a second probe that you make that hybridizes right next to the first probe on the messenger RNA, and it has a barcoded RNA segment on here, so that these are different colored dyes. And there are, I think, 6 or 8 dyes that you can use. So you can barcode, theoretically, 10,000 different probes by just altering the order of these guys. And that has a repeat sequence on the end of it as well. So the way that this works is that you put your capture sequences on the glass slide, and they just bind at random. And so you can have several hundred different capture probes on your glass slide. And each one will then you hybridize it with RNA. And it's done directly with RNA, and it's done in very small volumes. So you can use less than a microliter of RNA without having to do anything. You don't need to separate messenger RNA or anything. So it's really quite useful in that way. The messenger RNA binds to its cognate probe, and then you add on top of that your labeled probes for all of your genes of interest, and that will bind. So you'll end up with a bunch of different RNAs that look like this. You then put this slide under a slight electric current, and you wash it to get rid of all of the unbound probe. And you then hybridize with this binding probe that's complementary to this region here, and then that will bind both ends down on the slide. So now you have your RNA stretched out on the glass slide, and this probe has a series of different colored signals, fluorescent probes on it, and you can visualize the whole slide using a microscope camera, and you can count the number of molecules that are captured, and you can tell what they all are by looking at the order in which the probe occurs. And this is, of course, done all automated. You don't sit down and count them yourself. So you can end up getting a count of the number of molecules of each messenger RNA that's on this glass slide. So you count, essentially what you're doing is you're counting the number of molecules for each of those RNAs that were in the original RNA sample. And so you get a digital readout, and it's quite sensitive and quite reproducible. What you do need to know, of course, is you need to know what genes to look for, and then you need to buy the probes. And so it's expensive to buy the probes, but to actually do each sample, you can process them 96 samples at a time, and it only costs $100 or $200 to do 90-odd different samples. So the per sample cost is quite expensive. It costs money to buy these original genes. So to do a set of, let's say, 1,000 experiments is probably going to cost you $10,000 or something like that. But you generate the data really quickly once you've got it started.
You said that you don't need to isolate the mRNA. You can go straight with RNA.
You can go with total RNA, yes. Yes. And you can use very small amounts of it. And in fact, you can almost get down to single cell levels with Nanostring. So I think that's sort of a technique that holds a lot of promise in the future. And I've been asked by the Nanostring folks to try to get together a consortium of Leishmania people to buy these probes. So I'm not so sure we're quite ready to do that yet, but one of these days that might happen. The last technique I wanted to talk about was nuclear run-ons, which is actually measuring the transcription rate. And I won't go into any detail because no one in their right mind will do this in Leishmania. We did it, a postdoc in my lab did this a number of years ago, heroic effort. It only worked because he had really green fingers, so he could get any experiment to work, but it takes a lot of time and effort and you muck around with a lot of radioactivity. And the end result of that was that we found that everything is transcribed at the same rate in Leishmania, so no one needs to do it anymore. Although there is one experiment that might be sensible, and that is to really look at changes in transcription to see whether they're associated with different life cycle stages, because I suspect that there is some difference in transcription rates as you go between life cycle stages. And in fact, I think the available data would suggest that metacyclics don't really transcribe very much at all, although no one's ever done that experiment in a definitive way. Oh, and the final thing you can do is really not so much measuring levels of RNA, but you can use reporter systems that will give you very sensitive measures of enabling you to define regulatory elements, be they promoters in Leishmania probably, again, are not really terribly interesting to look at, but certainly regulatory elements that are in the 3' and 5' UTRs would be useful. So I'm mentioning all of those only so that you'll know what you have available. What I'm going to spend the rest of the time talking about is RNA-seq, which is really the one sort of in the middle that is the most modern system other than, I guess, Nanostring. So the way that RNA-seq works is that generally you take total RNA. You usually are going to be interested in messenger RNAs, so you do a messenger RNA purification to get rid of all of the ribosomal RNA. Otherwise, you end up with a lot of sequences that are just ribosomal RNA, which is not very interesting. There are kits available to do this in relatively small scale. So you purify your polyA plus RNA. You then either fragment it or do a reverse transcription with random probes. I can tell you that using random probes to prime your first strand in Leishmania is going to give you a very bad result, so go with fragmentation. And then you add on linkers, and usually it's going to be Illumina linkers, and you then generally have to amplify the library a little bit so that you end up with fragments that can then be sequenced. And there's really no necessity to sequence from both directions when you do RNA-seq, although nowadays it's just as cheap to do both directions as to do one direction, but you won't really get any more information. And again, you don't need to do long reads. In our experience, 36 nucleotide reads are sufficient to unequivocally map most reads to a single place in the Leishmania genome, but you can sequence 100 base pair reads if you wish. Okay, so again, I'm just telling you which platforms are available, and most RNA-seq is going to be done using the Illumina platform for reasons of cost and throughput. Now, there are several different ways that you can make your libraries, and you will use these to do different things, but you can make your libraries from fragmented messenger RNA, or you can specifically make them from small RNA that's already is small enough to sequence. And generally, you will add linkers to the 5' and 3' ends of the fragmented messenger RNA, and you will use those linker sequences to make your cDNAs. And as I said, that's preferable to random cDNA priming because of the GC richness of the Leishmania genome and the secondary structure, or probably the secondary structure of the messenger RNA means that when you use, quote, random hexamer primers to prime Leishmania messenger RNA, you end up with most of the reads in intergenic regions, which is not terribly useful for you. So don't do that. We fiddled around a long time using NSR, not so random primers, to mean that we didn't have to isolate messenger RNA, and eventually we gave up because it never really worked as well as it was supposed to. You can enrich for the 3' end of the messenger RNA by doing oligo-T priming on your first strand. So here, you're actually priming in the first strand, specifically off the polyA tail. And so when you then come back in the second strand, you're going to use a random primer in the second strand. So you're going to really bias your reads towards the 3' end of the RNA, which might be useful. In trypanosomatids, you have the big advantage that you can enrich for the 5' end of the messenger RNA by using a random primer in the first dimension, and it doesn't matter that it's really not random, but it's basically you just need to make first strand cDNA. And then you use a primer for your second strand synthesis that's complementary. Well, actually, it's the same as the splice leader sequence. And so you're going to then make cDNA fragments that are located at the 5' end of every messenger RNA. And so that is really useful in that you don't need to actually make polyA plus RNA because obviously the ribosomal RNA is not going to prime in the second strand. So that can be very useful where there's not really enough RNA to do a polyA plus priming. And I'll show you a couple of applications of that in a moment. The other use of this is that it will tell you precisely where the 5' splice site is, and you can look at differential splicing using that technique as well. The last technique is really a variant of this called ribosome profiling, which essentially starts with RNA that was isolated from polysomes. And what you're mapping here is you're mapping the region of the messenger RNA that's actually being translated at that given instant. And so by comparing that to libraries that were made from total RNA, you can look at differences in translation efficiency of different RNAs, and I'll talk about this at the end. Okay, so I have a few slides now that basically tells you what you have to do after you've gotten your sequence back from the sequencing lab. And so you're going to have 20 million reads from each of your libraries, and they'll be in FASTQ format, and you have to process them. So there's a very large bioinformatics pipeline that needs to be put into place to get this to work. So the first thing you need to do is quality check, and then you need to map them to the genome, and then you'll generally do a whole pile of analyses that will be different for different experiments that you're doing because you'll be wanting to ask different questions. And I'm going to go through these in a little bit of detail just to give you a sort of a flavor for what's involved. So the first thing you want to do is you want to make sure that your read quality is good. And so this is an example of an output that's generated using a special R script. And this is what a good read will look like. This is a 36 nucleotide read. And so this is measuring the quality here, and that's all should be up above 30. And this is measuring the base composition. And what you should do is you should see that you should get a random distribution of bases at each position that should mirror the base composition of the genome. So this would tell you that things look good. That's what a good one will look like. I also like to look at the 1,000 most frequent sequences. So basically, we have scripts that will go in and pull out all of the sequences that are identical and bin them, and then you will pull out the 1,000 most frequent, and we'll want to make sure that the vast majority of these aren't a single sequence. So if you have a ribosomal RNA contamination, all of the 1,000 most frequent will be ribosomal RNA, and so that's not going to be very useful. And so we can use this to filter out bad reads if we want to. Just a little bit more detail here, what a read looks like. And again, this I think is a 36 nucleotide read. So each short read is, as you can expect, a set of nucleotides with a discrete sequence, and each base gets a quality score, which tells you the likelihood that base that was called by the software is correct. And so it gets a quality score between 1 and 40, 40 being the best and 1 being really lousy. And so you want your quality score to always be above 30. The data will come back to you, as I said, in a FASTQ format, which will be a huge text file that looks like this. So you have 2 lines. For each sequence, you get 2 lines. You get the sequence, which is the DNA base that's been called by the program, and then underneath that, you'll get another line that gives you the quality score in an encrypted format. And in this particular case here, I means that this quality score was probably 40, which has a 1 in 10,000 chance of error. So that's a very good quality, whereas the dollar sign and the 4 have a slightly lower quality. And I haven't memorized these encrypted quality scores, but the software will read this for you, so you don't need to actually look at these. And in fact, these files are generally so large that most text readers won't actually open them. These files can be, 1 or 2 gigabytes in size, depending on how many reads you've got in there. So, as I said, this is a typical good read. Good quality, even out to base pair 34. Although the quality scores do go down as you go further out. Now reads typically are going to be out to 100, and you still should have reasonable quality out to there. These are very old data. Here's an example of a set of reads that looked like something bad happened. So here the quality looks good up until cycle number 41, and then something happened. Right? The quality scores all of a sudden fell off the map, and in fact the base composition did something strange at that position as well. And then out here the base composition is really weird because you've got a lot of ends being called in. And what happened in this particular instance was that, whoops, sorry. What happened in this particular instance was that the machine ran out of nucleotides in cycle 41, and so it didn't have enough nucleotides to continue out here. I had some other cute examples where we had a shutdown, the machine shut down overnight, and then when it resumed, it had forgotten where it was, and so it counted the base twice. So we ended up with sort of a duplicated base in there in all of our reads, and of course none of them would align with anything. So we ended up having to throw these away. Align the sequences to the genome. We like to use Bowtie 2 again, but you can use other tools as you want, and there's lots of parameters that you can tweak. And then there's a whole series of SAM tools that take this data and turn them into BAM files, which you can then use to visualize your alignments against the genome in your favorite visualization tool. I like to use Artemis myself, but there are other tools available. The alignment to the genome, as I said, you can change a lot of parameters, and you might want to use different parameters to do different things. You can use different parameters in different algorithms. You can trim the bases, the bad bases, or the early bases if you want. You can set different mismatch thresholds using different types of parameters. And so, depending on how rigid you are on setting your mismatch, if you set it too stringently, you'll miss some reads because the reads are not always perfect. If you set it too loosely, you'll end up with reads that map to too many places in the genome. You can also change how you report things. You might want to report only those reads that have unique maps, but then you're going to miss all of the repeated genes. You might want to decide that you want to count the same read more than one time. You want to map it to every place where it aligns, or you might want to, my favorite is to map it to the best, only once, map it to the best place, and if it can't decide which one is better, if there are several options that are all of the same, it assigns them to it randomly. So the optimal alignment is in fact an iterative process depending on your genome and what you're seeking to do. So you have to do your alignment, go see what you've got, and then change it again a little bit. Once you've settled onto a set of parameters that works, you can use that for all subsequent experiments unless you want to do something slightly differently. Oh, I think I'll just skip over this. This is telling about how you can deal with multi-matches. But essentially what you'll end up at the end of the day, you'll end up with a report that says X reads didn't match at all. And that probably tells you that these are reads that really weren't good enough to match. They had too many mistakes in them. Or B, that your reference sequence has gaps in it. Or C, that your RNA is contaminated with RNA from another organism. Right? So if you get a lot of non-hits, no hits, that tells you that something was wrong in one of those three things. Yeah, typically, you'll get around 70% of your reads matching. And if you get much fewer than that, then that's telling you that there's a problem. most of the reads should be unique hits, particularly in Leishmania, which doesn't have a lot of repetitive sequence. But you'll get about 10% of your reads will have multiple hits, either from 2 to K locations. And you might say that you don't want to look at things that have too many matches because it might just sort of be impossible to tell what they are. So you can throw them away if you want to. And so you can change all of these parameters and basically just move things around from one bin to another. And again, you want to choose the one that works best for you. This particular library here is what we're measuring here is we're measuring the percent of reads that didn't match the genomic sequence over its length. So you can see here that in this region here, in the center of the read, almost all of the reads matched somewhere in the genome. At the beginning of them, most of the reads didn't actually perfectly match the genomic location. And that was because in this particular library, the first 2 bases were a 2-base barcode that was common to all of the sequences, so only 1 in 4 of those will match the genomic sequence at that location. Then we had a random base here that, again, 1 in 4 will match. And then we had some bases here which were from the primer. And as they become further away from the priming site, they will, again, have mismatches. And then obviously you have some more mismatches up at this end. So you might choose, when you were doing an alignment, you might choose to trim all of these off so that you wouldn't have to worry about them having mismatches.
But then this will work well if you have a reference genome. But if you don't have a reference genome...
If you don't have a reference genome, obviously you can't do this, no. So what you can do is, if you don't have a reference genome, is you can treat your RNA reads as a genomic sequence, and you can do an assembly of your RNA reads, and you basically come up with an assembly of each individual messenger RNA. And then you can use that as a reference, right? So these days, it's generally, you're going to have a reference genome to align against. It doesn't have to be exactly the same strain. It just has to be something that's similar enough that you can guarantee that most of the reads will match somewhere in the reference. So for instance, for a long time there was no L. donovani reference genome, and we aligned our L. donovani RNA-seq reads to the L. infantum reference, and that worked just fine. I mean, we probably missed a few things, but most things they worked fine. So again, you know, L. major and L. tropica would work fine. L. brasiliensis would do all of the Vianias for the most part. So your reference doesn't need to be the exact same sequence that you have, same strain that you're working with. Okay, this was the example that I was talking at before. When we did this on this example here, what we find was that something really weird happened here at cycle number 41. No, that's actually not 41. It was cycle 33 here. At cycle 35, everything started to mismatch, right? So we were getting a lot of things that didn't really match properly. And when we repeated the analysis, someone had a bright idea that, oh, well, let's see what's the frequency of having the same base in 2 positions in a row, because you would expect that at random to only happen 25% of the cases. And so, in fact, that was correct most of the time. But on this cycle here, you can see that 80% of the reads had the same base as the base before. And so that was where we figured out that the sequencer had paused overnight because the disk drive had gotten full. It had generated too much data. Here's an example of, even before you did the alignment, you can look at your read here, and you can see, oh, this is really good high-quality sequence, got great quality scores, but I can actually read the nucleotide sequence here. And this is the average of all of the reads. This is not a single read. This is the average of all of the reads in this library. And there were 17 million reads, and almost all of them had the same sequence. So out of the 17,393,000 or 394,000 reads, 98% of them had the same sequence here. And of course, it was the smallest ribosomal RNA locus. So this library was not a particularly useful library. Although we could discard these 98% of the reads and just look at the others, and we would have a few tens of thousands left, but that's really not enough to do statistics on. So, if you saw something like this, you wouldn't bother doing any analysis on it. You would have blown the money for making the library and sequencing it, but nevertheless. So one way around it, actually, if you want to avoid that, is you sequence each library at a very low coverage on a MySeq machine and just get a couple of 100,000 reads, which is good enough for quality checking. Okay, after you've mapped it to the genome, then the next steps are really going to depend on what you want to do. Generally, what we'll do is we'll look manually to see whether they've mapped to the right strand. And then usually what we're interested in is we're interested in getting a count of the number of reads that are associated with each protein coding gene. So basically what you do is you count the number of reads in this position here. Now, this particular alignment here is showing something a little bit strange because you'll see that there really are very few genes that overlie the protein coding gene, and that's because this is a splice leader library, right? So the reads are all mapping to the 5' end of the messenger RNA, which is upstream of the gene. So to count the reads that are associated with this gene, we have to look at all of these reads and say, okay, this belongs to this gene. If this had been a fragmented RNA library, and I'll show you some later, the reads would be all spread across this whole region. So that's why I'm saying that you've got experiment-specific analyses. Now, I said that these different libraries can be used for different things. So here are some questions that you might want to ask in Leishmania. If you want to just map the transcripts, if you want to just find out where the ends of the transcripts are and whether there are other small RNAs that you don't know anything about, you might want to make a fragmented RNA library or a small RNA library. You can use splice leader priming to map the 5' ends, and you can use oligo to T priming to map the 3' ends. So 3 different libraries will give you 3 different pieces of information. If you wanted to measure, rather than just measuring where the transcripts were, you wanted to actually measure the changes in transcript abundance, you can do it with either of these 2 methods. I can tell you now that using fragmented messenger RNA is probably a little bit more reproducible than making splice leader libraries because there's fewer biases in making these libraries. But nevertheless, you can do it on splice leader RNA-seq libraries to use that to measure transcript abundance, and I'll show you a lot of data on that. Splice leader RNA sequence libraries are going to tell you about alternative splicing, and I'll show you a couple of examples of that in subsequent slides. And then ribosome profiling will not only measure the translation rate, but it'll also be useful correcting the annotation. And I'll show you some examples of that where about 5% of the gene predictions in TriTrypTB are wrong in terms of where their 5' end is and whether there's really a protein coding gene there or not. So I'll show you a couple of examples of that. We're going through and manually annotating both the T. brucei and the Leishmania donovani genome based on ribosome profiling, so we can expect that data to start coming through into TriTrypTB over the next few months. So made a fair number of libraries on a fair number of organisms. I think we've made almost 200 different libraries now, so this slide is a little bit old. From 6 different species. I made polyA libraries only from 2 different species, from L. major and from L. donovani, because basically I decided that it was a lot of work for not very much results. But I was interested in finding out whether it would work and actually mapping the 3' ends in these species quickly. And then we made splice leader libraries really from all of these. I've now started to make more fragmented libraries as well. So you really use 3 different types of libraries. But initially, I was making splice leader libraries. And you can see that we covered almost all of the genes. We got, we got reads from more than 80% of, or 90% of the genes with the splice leader library. you get sort of different patterns when you look at the splice leader and the polyA plus libraries. The green bars are from the splice leader library. This is, I think, from L major. And the red bars are from the polyA library. And what I'm mapping here is I'm mapping how many reads mapped to each gene. And so here, there were only 10 to 20 reads that mapped to these couple of, 50 genes had only 10 or 20 reads. Here, there were 20 to 50 reads. So a couple of hundred genes had 20 to 50 reads each. And then all the way out here, we had a few genes that had 10,000 reads or more. So you can see that there's a big change in dynamic range of this. The splice leader libraries generally were better, so you get more reads per gene from the splice leader libraries than the polyA plus libraries. But again, we got high coverage. So almost, so more than 80% of the genes had at least 100 reads. So that means you can do good statistics. If you have 100 reads and you compare two different libraries, you can get good statistics on that. If you only have 5 reads, comparing 4 in one and 6 in the other is not going to be terribly robust. Most genes have only one major splice leader site. This is a sort of a difficult chart to interpret. Well, what we've got here is we're on this axis here. We're mapping the percentage of the reads that mapped to the major splice leader site. So most genes we actually found had more than one splice leader site. But for most genes, one of those sites predominated. And in fact, for most genes, or more than a quarter of all of the genes, more than 90% of the reads mapped to a single splice leader site. But there were a number of genes, here for instance, where the major site only accounted for 50 to 60% of the reads. And so there were probably 2 sites, at least, for most of these genes. There were 2 sites that were being used in relatively similar amounts. You can contrast that to the polyA sites, where very few genes had a single polyA site. Almost all of the genes had 2 or 3 different polyA sites. And so splice leader addition is much more directed than polyA site addition. When we looked at the position of the splice site and the polyA site relative to the annotated CDS, we came up with maps that looked like this. So for splice leaders, the splice leader site was generally within the first 500 base pairs upstream of the annotated start site of the gene. Although in some cases, the major splice leader site was further out. Some cases, the nearest one was more than 3,000 nucleotides away. About 5% of the time, the splice leader site, the major splice leader site, was actually downstream of the ATG. And as we'll see in a moment, that's almost certainly because we got the open reading frame wrong. What we generally did was we generally called the longest open reading frame, that is the first ATG in the open reading frame, and in about 5% of the cases, that's actually not used. So obviously you can't translate this gene into that protein because the RNA doesn't go out far enough. The 3' UTIRs were usually a little bit longer, so you can see there's a lot more of them that are in the 500 to 1,000 base pair range. And here, about 10% of the time, we found polyA sites that mapped within the CDS. I'm not sure whether this is largely an artifact, because the polyA priming, the oligo to T priming will sometimes prime at A tracks that are within the coding sequence. That's fairly infrequent, but nevertheless it does occur. But there is some evidence that in a number of cases you can get polyA sites before the end of the CDS. And obviously that messenger RNA is not going to be translated. So it's probably starting to be degraded. So we can use this information to start with to re-annotate the genome. And so one of the things that we did in L major when we first got this data is we developed a set of scripts that would predict from the splice leader and the polyA sites where the splice leader site and where the polyA site was for each gene. And then we developed a graphical interface to view those results. And we went through and we looked at them, all of them individually by hand or by eye, to see whether the predicted splice leader site and polyA site actually was correct or not. So here's an example here. This is what the viewer looked like. This is the protein coding sequence. The blue tells you where the splice leader site is, or the splice leader reads are mapping. The height of the bar tells you how many reads there are. The red tells you where the polyA reads are mapping. And so here, it's clear that the splice leader site is here, and the polyA site is there. And I think for this example, that's actually where the algorithm had called them, so we didn't need to correct it. We ended up having to correct about 10% of them. And so this data is actually in GeneDB now, I think, and should end up in TriTripDB before too long. We've actually had this data for about 2 years, I think, and it's been delayed getting it into GeneDB because they didn't have enough curators at the Sanger to do that. Okay, I already said that. Okay, here's an example that I was pointing out where we had to correct our gene model. So this particular gene here, if you look at L major and L infantum, the open reading frames of the predicted coding sequences are quite a bit longer. They're 95 base pairs longer for L major and L infantum than they are for L brasiliensis and L mexicana, right? And when you do the alignment, you can see that they align really. Those 4 species have a very high degree of sequence conservation here. So it was a bit strange that these 2 genes would be different. And you'll notice that they don't start at the same position either. So when we did the splice leader mapping, what we found was that in L infantum and L major, the splice sites mapped within the coding sequence. And so clearly, this region here was not going to be translated. Whereas in L brasiliensis and L mexicana, which I'm not showing, the splice sites were mapping upstream of this sequence here. So in fact, in all 4 species, the splice site was right around about here. So obviously, we had gotten the models wrong in L infantum and L major, and we had to correct them. And this was the true start of the protein coding sequence in all 4 species. If you look at the existing genomes, you will find hundreds of examples of this. So there are hundreds of genes in the Leishmania genomes that are on TriTripDB now where the splice site is where the beginning of the translation is incorrect. Most of the time, it will be too long, but sometimes we actually made the wrong choice going the other way. So that needs to be corrected and is in the process of being corrected. So my warning to you is when you first start working on a new gene, you should find out where the translation really starts so that you really know where the 5' end of the gene is, rather than assuming that the sequence in TriTripDB is correct, because about 5% of the time it'll be wrong. Hopefully that will be fixed as we go by, but L major should be in the database soon. L Donovani and L infantum should be corrected relatively soon. The other ones will have to wait. The other thing that we found was novel transcripts. So here's an example of a gene that incidentally has an internal CDS, so it was called incorrectly. But immediately upstream for that, we see a whole bunch of splice leader reads here and a polyA site here. And so what we find is that there's likely to be a transcript that goes from this splice site to that polyA site that doesn't have any gene associated with it. So there's probably no long open reading frame in here, otherwise it would have been called as a gene. So we would say this is a non-coding RNA. We'd have to go in and look at the ribosome profiling data to see whether there really is any translation of this gene. So it could be that there's just a short open reading frame there that is translated and we got it wrong. Either that or there could be a frame shift. So we'd have to look at some more data. But anyway, it turns out that there are several thousand non-coding RNAs distributed throughout the genome. And in fact, in Leishmania and African trypanosomes, virtually the entire genome is transcribed and has stable RNAs associated with it. So whether these have any function or whether they're just hanging around because they happen to be processed and are relatively stable is unclear. Okay, so now I have a couple of examples of how we've used this data to answer biological questions. So I have 5 of them, actually. So I should remind you what base J is. It was named after someone in Pete Boerst's lab, Janet Gomez-Omt. So that's why it's called base J, because her first name started with J. And she was the one who identified what the structure was. About 1% of Ts in trypanosomatids and euglena are modified to make J. So J is a glucose molecule that's hooked on to the hydroxylated thymidine. In Leishmania, as in other organisms, most J is telomeric, and interestingly enough, only the second T of the telomeric hexamer sequence on this strand is turned into J, whereas the single T on the other strand is made into J. A couple of years ago, in collaboration with Pete's lab, we mapped where all of the J's are in the Leishmania genome, and they turned out to be found at convergent strand switch regions and at what we thought to be internal transcription termination sites. And so all of the convergent strand switch regions, with one exception, had J peaks, and all of the internal transcription termination reinitiation sites had J peaks. About a third of the transcription start sites at at divergent strand switch regions had J, but the other 2/3 didn't. So what Pete's lab had done was they had knocked out one of the enzymes involved in J biosynthesis, and they'd shown that if you knock out JBP2, that causes the cells to lose most of their J. The cells actually grow quite normally. And the J is lost slowly over time. And the reason for that is that the remaining enzyme, JBP1, is involved in maintaining J, but it can't add J de novo. JBP2 is needed to add J de novo, and then JBP1 keeps it there. But that's a different story that I won't bother telling you about. The internal J is lost more quickly than the telomeric J in these mutants. So what did that do to transcription? We used RNA-seq of both small RNAs and also splice leader RNA-seq. 2 different libraries were made from wild-type cells here. And when you look at the sort of chromosome scale, what you find is that in wild-type cells, there are very few antisense transcripts, particularly in the small RNA. So what we're measuring here is the green lines indicate the number of reads that map to each of these positions, and the same here. The red lines are where it's gone off scale, so we had more than 50 reads. And so you'll find that on here, these are the genes on the top strand. You see very, very few reads on the opposite strand. So essentially what that meant was there's very little antisense transcripts present in stable RNA in wild-type cells. When you looked at the J2 knockouts, what we see right here is we see a lot of read-through transcription at this convergent strand switch region here. So we've now got a lot of reads that are mapping to the antisense strand. And you'll also note that there's a gradation. So the reads fall off. The further you go past this site here, the fewer reads you get. So it looks like what's happening is that these cells no longer terminate transcription properly. So the conclusion was that base J is needed for transcription termination in Leishmania. There was an example, I don't have a slide for it. I remember I said one convergent strand switch region did not have base J in wild-type cells, and at that site there was antisense transcripts in the wild-type cells, in both L major and in L tarentuli and in L donovani, but not in L brasiliensis. because L. brasiliensis, presumably because L. brasiliensis has RNAi, and so doesn't like to have double-stranded RNA. All right, so there was a question as to why IJ is lethal. If we lose all of our J, and the way we did that was we took the J2 knockouts and we added bromodeoxyuridine, and they lost all of their J essentially, and the cells died. What we did was by counting the number of sense reads and antisense reads, we were able to get clues as to what might be going on here. And what we found was that there was an increase in the number of sense reads in places that looked like this, where we've got a transcription stop site and then a reinitiation site. We got increased abundance of some RNAs, and then we got decreased abundance in some other RNAs. And so we still don't know why loss of J is lethal, but it could either be because they're overexpressing a toxic gene or they're underexpressing an essential gene. So that remains to be seen. All right, second set of experiments, and this has been published, was a study that we did in collaboration with Norma Andrews in L. amazonensis. So she grew amazonensis for 24 hours in the absence of iron. Is there a question? No. We made splicellida RNA-seq libraries from cells that were grown with iron and compared them to those that were grown without iron. And we aligned the sequences against the Mexicana genome because amazonensis hadn't been sequenced, but that worked just fine. And what we found was that there were only 68 genes that were upregulated by more than twofold. And we did these in duplicate, so the data are quite solid. And these were genes that were involved in ion transport and autophagy. So a lot of that made sense. And then there were another 66 genes that were downregulated. And these were genes that were involved in electron transport, and many of them had iron cofactors. So again, that made sense. So basically, if the cells are starved for iron, they upregulate their transporters to get more iron. They also start to upregulate autophagy genes because they start to kill themselves. But they also downregulate the expression of proteins that need iron. But it's a very small number. I mean, it's only 60 genes up and 60 genes down. So this really sort of, I think, was a nice way of finding this. Norma had already shown that these two genes here had been upregulated in the absence of iron. So in one experiment, we could increase from 2 genes to 100-odd genes that did various interesting things. And luckily, this turned out to be a nice sort of logical story, although there's a lot of hypotheticals in here as well. So the assumption is that many of these hypothetical proteins probably have iron cofactors over here, and that may be involved in iron transport here as well, or autophagy. We did a similar experiment with Nicola Carter, where she starved the cells for purines. We made libraries again. And here, we only did the experiment once, so the numbers are not as robust. And so we got a lot more genes that were up. But again, transport genes are up. For some reason, one of the GP63s went up. And again, they started to feel unhealthy, probably, so that it upregulated their autophagy genes. And then there were a few genes that went down. These didn't make a whole lot of sense. They looked mostly to be hypothetical proteins. But maybe some of these make sense. Here's a kinetoplast DNA-associated protein. If you don't have purines, maybe you don't need as much of this protein, et cetera. So we need to repeat this experiment to see whether we can get more tight statistical data. One obvious experiment to do is to look at stage-specific expression. So here's an experiment that we did with Steve Beverly in L major. We compared procyclics, metacyclics, and amastigotes. This experiment has been done in triplicate. This was just, I think, when we'd only done one experiment. So if you compare, in this particular experiment here, if we compare promastigotes to, what am I comparing them to, amastigotes, we see that about 1,000 genes are upregulated in promastigotes by more than twofold, and about 600 are upregulated in amastigotes. So a fairly big change. And if you look at the amastigote-specific proteins, and I don't know who can read this, I can, but all of these guys are amastins. So that's to be expected. I think if I go back a slide here, if I look here, these are things that are upregulated in promastigotes, and we find paraflagellar rod proteins. So many of these things made sense. Interestingly, there's some histones are upregulated and tubulins, and that's because promastigotes grow faster than amastigotes. So fairly large numbers of genes change. What we also find was that for several hundred genes, there was a change in the position of the major splice leader site. And I'll come back to that again in a moment. So there's stage-specific regulation of the major splicing site. When we looked at metacyclics, there were even more genes that were different. So metacyclics, in terms of their their RNA levels, are actually less similar to promastigotes and amastigotes than they are. They're less similar to promastigotes than amastigotes are similar to promastigotes. And that's because they've stopped growing, and so their RNA levels change a lot. So that goes back to a point that Greg made before, is that the growth state of your parasite really makes a big difference when you're doing these experiments. It's even more so when we look at translation. So if you want to get reproducible results, you have to make sure that your cells are growing the same way, or you're going to be confounded by changes in growth rate. Now that's an interesting perspective in experiment in and of itself. Okay. When we were doing these experiments with Steve, we decided that, okay, let's look at what happens with amastigotes. He had made his amastigote RNA from amastigotes that had been isolated from footpad lesions, because you can't get axenic amastigotes from L major. So that's a fair amount of effort to do that. So what I decided to do was, okay, well, why don't you just mash up the footpads and maybe try a very, very quick procedure for isolating amastigotes, and we'll try to make our splice leader libraries off that. And we did that, and we got really quite good libraries, 20 million reads. And we got very similar results from this experiment as we did using the isolated amastigotes, with only 50 genes that showed very much difference. And so basically this says that you don't need to purify your parasites, let alone purify the RNA. All of this is done on total RNA, by the way, because it's a splice leader library. So the other thing that it says is that mucking around with the cells and isolating them probably didn't change them all that much, because not too many genes changed. But there are some genes that change, so maybe these are sensitive to cold or something like that. I haven't really looked at them. Here's an experiment we did with Greg's group, looking at the difference between axenic amastigotes and intracellular amastigotes from L. donovani. As most of you know, there was debate for some time about whether axenic amastigotes were just messed up procyclics, or whether they were amastigotes. And Barbara Papadopoulou's lab had published some microarray data saying that they were quite different. So we decided to test that experiment using RNA-seq. And so we took Wenwei in Greg's lab, isolated RNA from axenic amastigotes and from amastigotes that were growing inside macrophages. So here he didn't actually isolate the amastigotes out of the macrophages. He just made total RNA from infected macrophages. And we made libraries from both of those, and we compared the two. And so you can see that there are only 7 genes that showed a 4-fold or more change up, and 7 genes that were down. So essentially, these two, axenic and intracellular amastigotes, looked virtually identical. The genes that were changed were transporters, which makes sense because the cells are growing in different environments, and then a couple of lipid metabolism genes, and again, they probably have different lipid precursors if they're growing in a macrophage than they are in a culture, and a couple of peptidases, and then hypotheticals. So there were some differences, but not so many. Here's an experiment for Jesus. So with Pedro Alcuila and Vicente Laraga and Ricardo Molina, Pedro had isolated RNA from promastigotes that had been obtained from sandflies, and we compared them to promastigotes that had been cultured for 5 days. So these were from the anterior gut of Promiscuous, I can't even say that. This particular perniciosis. I don't remember what day these were from. When we compared the two, there were a lot of differences.
More than the amastigote?
Many more differences between axenic promastigotes. Remember, these are axenic promastigotes. They're not in their natural environment. These are sort of late logs, so these would be procyclic promastigotes. Comparing these to things in the anterior gut, which should be sort of what you would think of as procyclics, but there were a lot of differences. Okay, but so I think this is, of interest, saying that in fact the cultured promastigotes look less like insect-derived promastigotes than axenic amastigotes look like macrophage-derived amastigotes. So the experiment that I'm looking forward to doing with Jesus is to look at different types of parasites from different regions of the sandfly gut, because my bet is that they will show significant and substantial differences.
And that will also, we may understand metacyclogenesis.
Exactly. So we know that when you look at axenic metacyclics, they are really quite different to procyclic promastigotes. Many of the differences appear to be differences involved in stationary phase. So it'll be interesting to do that experiment. We're actually, Ricardo was trying to repeat this experiment, so it'll be interesting to compare what happens in different parasites in different sandflies and see what's similar and what's different. Okay, the next story I want to tell you about is something I've been working on with Dan Zilberstein for a number of years, and that is the differentiation process going from promastigotes to amastigotes. And Dan's worked this out, how to do this in culture, in axenic conditions, by raising the pH and lowering the temperature. And what he found was that there were really 4 distinct phases of differentiation that occurred over a 4 or 5 day period. Within the first 5 hours, the cells stopped growing, and in the first, what he calls phase 1, the first 5 hours, is basically perceiving the signal and responding to the signal. So they don't look all that different for the first 5 hours, and the RNA levels, I'll tell you, doesn't look very much, but there's a lot of phosphorylation changes going on during this time frame. By 5 hours, they've stopped growing and they start to bunch up. So in this stage here, phase 2, they basically go into a growth arrest and do some morphological changes, and they start to lose their flagellum. So then in phase 3, they're maturing and they start to grow again. So they've now sort of reorganized their genome or their transcriptome or their proteome, and they now are starting to grow as amastigotes. But even at 24 hours, when they look, for all intents and purposes, like amastigotes, there's no morphological changes during this time. There's another phase 4 here where there are a lot of changes in their metabolism. So there are really 4 phases. So what Dan did is he took samples at different time periods during this and did measured the RNA, firstly by microarrays, and then by RNA-seq. And Dan measured the protein levels or changes in protein using iTracker in the same samples. And we did this in duplicates. So this was data that was published earlier this year. If you look at the proteins, there's a number of changes in protein levels. They were able to detect 1,700 proteins out of the total 8,500. So it's reasonable coverage, but obviously not complete coverage. And of those, about 300 of them showed at least a 2-fold change. More of them went down than went up. And most of the changes in protein level occurred in phase 3. So there really was not very many big changes in protein level in phase 1 and 2. There were some. For instance, the paraflagellar rod proteins went down, as one might expect. But most of the changes happened in phase 3. or began in phase 3 even. When you looked at what was happening here, the amastigotes had downregulated glucose metabolism, and they downregulated protein synthesis, and they'd upregulated beta oxidation and amino acid catabolism and their TCA cycle enzymes. And so they basically had, as Dan put it, retooled their metabolism. So if you look on this map here, here are the things that are higher in promastigotes. Basically, only glucose metabolism is higher in promastigotes, whereas a whole pile of things in red here are higher in amastigotes. All right, so that's what happens at the protein level. When we looked at the RNA level, we saw a sort of similar picture, and this was done by microarrays. We saw several hundred genes, because now we have essentially complete coverage. We had several hundred genes that, where their RNAs went up, and we saw more genes that went up, more genes that went down than went up, and that agreed with the proteomics data. The difference was that most of the changes occurred in phase 1 and in phase 2. So there were very few changes in RNA levels that happened only in phase 3 and 4. So the RNA changes seem to occur earlier than the protein level changes. Here's a list of the genes that we found changed, and I won't go through that. You can read that. It's all published as well. So before we went and looked at, actually, we didn't do this before, but what I should tell you is that in other systems, in high eukaryotes, studies now are finding that changes in translation are accounting for more than 50% of the changes in protein level. So really looking at the RNA only shows you less than half of the story. And this happens both in yeast and in mammal cells. So there's really not a very good relationship between RNA level and protein levels, is the take-home message in other cells. So we did the same thing in Leishmania using the data that I had previously shown you, and you can see that the correlation between the change in the RNA level, which is on this axis, I think, and the change in the protein level on that axis is really pretty bad. It's moderate. This is 2.5, 5, 10, 15, 24, and 120. It's pretty good in this time, but by the time you get to amastigotes, the R-squared value is really bad. So there's a very poor correlation between changes in RNA level and changes in protein level. So using changes in RNA level can be misleading. It doesn't mean that it's useless, but you have to be careful. And I'll show you in the next couple of slides what I mean by that. So here's a group of genes. So we had 902 genes that we had both RNA data for and complete protein data. So we had 902 genes that we knew what happened to the RNA and what happened to the protein at all of these time points. A third of those, the RNA went down and the protein went down. So about a third of them. And then we had another 90 where the RNA went up and the protein went up. So about just a little bit less than half of all of these genes, the RNA and the protein levels went in the same direction. We had another 198 genes where neither of them changed. So maybe 2/3 of the genes, the RNA and the protein agreed reasonably well. Not perfectly, but reasonably well. But we had 66 genes where the protein level went down, but the RNA level actually went up. We had another 132 genes where the protein level went up and the RNA level went down. So 200 genes where the RNA and the protein went in opposite directions. And then we had another 115 genes where the RNA level went down, but the protein level didn't change. So a number of cases where you get the wrong answer by looking at the RNA. So that was done with microarray data. We went back and we redid it with splice leader RNA-seq data, and we got essentially the same result. It was done on the same RNAs, except that we got more RNAs that showed twofold or more changes. Interestingly, when we did it here, we got more genes that went up than went down. That's the opposite of what we got when we did it at the microarray level. And I think at least part of the answer for this is the way that the data are normalized. Because normalization is actually never done correctly, mostly because you can't actually normalize correctly, because what are you going to normalize against? Most people normalize against total RNA. Well, we don't know, and in fact it's quite likely that the amount of RNA in a promastigote is different from the amount of RNA in an amastigote. So if you're looking at a per cell basis, your normalization won't tell you that. Not only that, but there are also differences depending on the abundance of the RNA. So RNAs that are expressed at a very low level tend to not change as much as those that are expressed at a high level when the cells go into a slower growth rate. So it's also, how do you normalize for that? So you can't, and you could say, well, why don't you normalize to a housekeeping gene? Well, my answer to that is tell me what a housekeeping gene is and show me that it doesn't change. So essentially what we're doing here is we're normalizing against the median RNA level as measured by RNA-seq. And so I think that that probably explains part of this. And now the other possibility is that what happens is that we're seeing a lot more changes at the 5' end of the RNA than in the RNA abundance. And so this is sort of telling us that something different is happening at the 5' end than the total RNA. But anyway, I haven't sorted that out. What we did find, though, was that there was a lot more downregulation in phase 1 and 2. So in fact, the phase 1 and 2 results mirrored what we got with the microarray results and with the protein results. What we're seeing here is that in phases 3 and 4, that's where we're seeing a lot of RNAs that appeared to go up. But I think that's actually an artifact of normalization. And that's perhaps brought home by this experiment here, where we compared the results from the microarray versus the RNA-seq data, and we find relatively modest correlation coefficients. And it gets worse the further out you go. What it does point out, though, is that there are a bunch of genes where by microarray we saw no change in RNA levels, but by splice leader we did see substantial changes. And I think what that's telling us is that these are genes where the protein coding sequence is very similar. So when you do the microarray, you're averaging the level of several different genes. Whereas when we do splice leader, RNA-seq, we're able to distinguish between those two different copies. And one of the copies will change, but the other one won't. And so it will be masked in the microarray results. So you get more sensitive results. Also, the fold change is generally higher by microarray, by RNA-seq as well. All right, so we did identify one set of interesting things from the splice leader RNA-seq that you wouldn't get from a normal fragmented RNA-seq library, and that is by measuring the splice leader site, you can see some genes, and in fact most genes, where there was a change in abundance, and they used the same splice leader site in all cases. So here you can see this particular gene, you get a lot more reads at 20 hours, so at 10 hours. So it seems to go up in abundance and then come back down again. So it's transiently upregulated. There are some genes where what happens is that you get no change in the total number of reads, but you're changing the splice site that's used. So in this case here, in promastigotes, both of these sites are used at about equal abundance. In fact, splice site 2, this one here, is actually used more than splice site 1. But by 5 hours, almost all of the RNAs are using splice site 1. So you've got a change in the splice site, but no change in the total RNA level. And then, of course, there's several genes where both the splice site choice and the total number of reads change. So these guys could be very interesting. So we found 2 examples. Actually, we found four or five examples where we had protein data and RNA-seq data. And so we were able to see, does the change in the splice leader site usage have an effect on the protein data? In fact, here's 2 examples where I'll show you that it does. So in this particular example here, this is a protein phosphatase, you can see that the RNA-seq data tell us that we go in promastigotes from a splice leader site that's that's further away from the ATG to one that's closer into the ATG. But you'll notice that the total number of reads here doesn't change very much, and the microarray gave us essentially the same answer. So there's really very little change in the total RNA level, but you're going from a splice leader site that's further away and moving into one that's closer to the ATG. And so what happens to the protein levels? Well, you get a decrease in the protein level. So apparently, by moving the splice leader site in closer, you get less protein being made. It's just a correlation, but I think that would probably indicate that what's happening is that there may very well be regulatory elements in this region here that are needed to get more efficient translation of that particular gene, right? And just in case you think that's actually the rule, let's go to the next gene, where the same thing happens. The total RNA levels stay the same across the differentiation. And here we're going from a more distal splice site to a more proximal splice site. And about the same. You're moving it in by a couple of hundred nucleotides. And in this case, what's happening is that the protein goes up. So here, making the 5' UTR shorter results in increased translation, presumably. So it is, I guess, formally possible that this change in protein is happening post-translationally, and it really has just a fluke that the splice leader site changes. But obviously, that's something that we would need to test. It seems sort of unlikely that you would find that, though. So what this is telling us is that it's quite likely that the parasites can use splicing to regulate the expression of a gene. So they differentially splice a gene under different conditions, and that causes the gene to be expressed differently, even though the messenger RNA levels don't change. Okay, so here, what's ribosome profiling? And basically what you do in ribosome profiling is you freeze your cells while they're actively growing, and you add, before you freeze them, you freeze their polysomes, their ribosomes, so that they don't translate any more RNA. And then you isolate polysomes, or isolate just ribosomes, from the cells after they've been flash frozen. So you've now got RNA that's got polysomes, or ribosomes bound to it. You then treat the cells with ribonuclease, and you have to titrate to get the right concentration of ribonuclease. And what that will do is it will digest all of the RNA except that 28 nucleotides that's protected by a ribosome. So you end up with a whole pile of 26 to 28 nucleotide fragments that were protected by the ribosomes. You then make an RNA sequencing library from that, and you use a god-awful procedure that takes a lot of work that has no bias involved in it, or at least as little bias as you can. Whether that's important or not is not clear to me, but we did it anyway. You then make this is called a ribosome profiling library. At the same time, you take total RNA from, actually take polyA plus RNA from these cells. So you use a little bit of these cells to make an RNA prep, and then you make a fragmented RNA library from their messenger RNA. And so now you have two libraries. You have a ribosome profiling library, which tells you what's being translated, and you have a library that tells you what the total messenger RNA concentration is. You map all of these through the genome. You actually make splice leader RNA-seq libraries as well, so that you know where all of the splice leader sites are. You could make a polyA plus library if you wanted to, but we didn't. Run this through the pipeline, align them all against the L-infantum genome, do a whole pile of other manipulation here so that we can separate these and do differential expression analysis, and you have to normalize the read counts, and you do it for all of these libraries. And then we end up with giant spreadsheets that we need to go in and make do number crunching on. Okay, so this is the data that you get. It looks like this when you look at in Artemis Viewer. So the light green are the reads that come from messenger RNA. And so you'll see for this 3 genes here, we see that this gene here, we've got a very small amount of reads spread out over this entire gene and going sort of a fair way upstream, and then a little way downstream. So the messenger RNA essentially is randomly distributed over the entire messenger RNA. So this gene here is more obvious. You can see that there's RNA across this whole region here, and then there's a little space here, and then there's more RNA here. So this is the RNA from this gene, this is the messenger RNA for that gene, and that's the messenger RNA for that gene. Then in dark green are the ribosome profiling reads. And so it's most obvious here. See that there's messenger RNA all the way across here. So there's a messenger RNA that probably starts here and goes all the way across to here. You only see ribosome profiling reads over the CDS. So essentially what's happening is you start translating here, and then you're looking at the so you have protection of the RNA as the RNA is being translated, and then the ribosomes drop off when they get to the end of the coding sequence. What you'll notice is that there's not a particularly good correlation between the RNA level and the ribosome profiling level. So this gene here has a lot of RNA, but really doesn't get translated all that well. This gene here has not very much RNA and gets translated a lot more. So by dividing the number of reads in the ribosome profiling library by the number of reads in the messenger RNA library, you get a measurement of the translational efficiency. And you can normalize that so that the median is 1. And so you'll get things that are, you'll get a measure of how well each gene is translated. And here, because of the way the libraries were made in a non-biased fashion, you can really be more confident of those numbers. So you can tell which genes are translated highly and which genes are translated lowly. And so here's some sort of typical results for that. Just looking at 0 hours versus 24 hours. This is the old Donovani differentiation story again. We've done an entire study on this in T. brucei. We worked it out in T. brucei, and that paper actually just came out this week in GMC Biology. But this is unpublished data from El Donovani. And so if you look at this particular gene here, if you look here at the messenger RNA levels between 0 hours and 24 hours, you'll see that there's really very little change. There's a small increase in the amount of RNA, of messenger RNA at 24 hours, but really it's maybe a couple of fold at most. Whereas at 0 hours, there's essentially no translation of this gene. 24 hours, it's really becoming more abundantly translated than any of the other genes. So there's an enormous translational regulation of this gene, which you would have really at least underestimated and probably missed if you just looked at the RNA level. So we find, I think in the next next slide I'll show we find hundreds or thousands of examples of that. In this particular example, interestingly enough, it looks like there's changes in the splice leader site. So here you see there's really no splice leader sites here. The splice leader site is way out here. So there's a big long RNA that gets that goes from here all the way over off the screen. Here, it looks like you've got splice leader sites that have come in and processed this RNA so that we now have what appears to be a discrete messenger RNA out of the middle of this big transcript. And that corresponds to an increase in translation here. So basically what's probably happening is that this precursor RNA is not being translated because it doesn't load onto the ribosome very well. When you change the splice leader site to here and you make the 5' end closer, you can now translate it. So that's one example. There are other examples where we see no, in fact, most of the time we see no difference in the splice leader site. So when we look at these results, we see that about 1,500 genes change their RNA levels. And here again, we're back to more down than up. So here's a non-biased, fragmented RNA, and it's giving us essentially the same results we saw with the microarray, and a different result than we saw with the splice leader library. So that makes me think that something is a bit funny about the splice leader library. 2,200 genes change their translation level. So there's another 700 genes that we would not have picked as being changed in their expression by looking at translation. And in fact, when you look at the change in translational efficiency, there are 2,400 genes that change their translational efficiency. And more of those actually increase their translational efficiency than decrease their translation efficiency, although it's not all that much different. And you can see that some of these differences are really quite substantial. We're seeing 8-fold or 16-fold or even 100-fold changes in translation efficiency. Whereas the RNA level changes are really quite modest. It's very few change by more than 8-fold.
Is there anything you can see different between the ones that are translated efficiently and the ones that aren't? In terms of splice leader sequence or sequence around there? Or?
Haven't really looked yet. Certainly haven't looked in this data. Elton has been looking in the T. brucei data and hasn't found anything obvious yet. We do have in the T. brucei data, and actually it's mirrored in this, that the translational machinery itself is very specific, is that normally the ribosomal proteins and the translation factors have high RNA levels, but only relatively modest translation levels. And that translation is exquisitely sensitive to growth rate. So it looks like the translational machinery has always got high RNA levels. I mean, you know, if you look in RNA-seq libraries that most of the most abundant RNAs are translational machinery genes. But in cells that aren't growing very fast, those ribosomal proteins and translation factors are not being translated. So it looks like the way that the cell works is that when they slow down their replication rate, the first thing that happens is that they stop translating their ribosomal apparatus, their protein synthesis machinery. And of course, that after a few hours will mean that there will be fewer ribosomes around, which will then mean that they then downregulate all of their other translation. So they use it looks like it's not regulated by RNA level per se. It's regulated by sequestering, probably by sequestering the untranslated RNAs and keeping them away from the ribosome. At least that's how it works in other cells. So in this system here, whoops, oh dear. What we did find was that in fact there was a global downregulation of translation during differentiation. So if you look in promastigotes, 68% of the polysome of the RNA was found on polysomes, where only 17% of it in amastigotes was found on polysomes. So there's a downregulation globally of translation. But some genes go the opposite way. So summarizing, I won't bother reading these. Lessons learned from RNA-seq. I mean, probably the most important one is you need to use a different method depending on the question that you're asking. And I should thank all of the people involved with that. So these are all people from my lab and Dan's lab, many of whom have moved on. And then a whole pile of different collaborators who've done various things together with us. And so that's RNA-seq.